Welcome, curious minds. Today we're plunging into something really fundamental to life itself. How animals, from the tiniest worm to the tallest giraffe, move vital stuff around their bodies.
It's all about transport.
Exactly. We've got a fantastic collection of sources for this deep dive, mostly pulling from Animal Physiology, from genes to organisms, second edition. And our goal, like always, is to pull at the key insights, give you those aha moments, and, well, make complex biology clear and hopefully fascinating.
Yeah, and you'll see how organisms really push against the limits of physics, how just one gene can change a whole heart, and even what a spider's jump has in common with, well, with human physiology.
Okay, intriguing. Let's get into it. So first things first, why do animals even need a circulatory system? Why can't things just, move around on their own? Diffusion.
Well, the surprising thing is just how incredibly slow diffusion is, over any real distance, I mean.
Yeah.
Think about it, a glucose molecule. It can zip across a tiny membrane in microseconds.
Okay, fast on a small scale.
Right. But for that same molecule to diffuse just 1 meter, you're looking at something like 20 years.
20 years. Seriously, that's astounding.
It really is. So for single cells or super small animals, diffusion works okay. Distances are tiny, metabolic needs are low. Even flatworms, which can be long, are thin enough that a branched gut gets nutrients close enough.
And as animals got bigger.
Exactly. Bigger, thicker, more active, higher metabolisms. Diffusion just can't keep up. The solution is what we call bulk transport.
Where the whole fluid moves.
Precisely. The fluid itself, carrying all the molecules and cells, moves much, much faster than diffusion alone could ever manage.
Okay. And to make that happen, these circulatory systems usually have, what, 3 core parts?
Typically, yes. You've got the fluid itself, blood or hemolymph, carrying gases, nutrients, waste, hormones, even heat. Heat's crucial, especially for us mammals and birds, but some reptiles use it too.
Right. Got to move that heat around.
Then you need a pump, usually a heart. And third, the vessels, the vascular network, the pipes, basically.
Like a city water supply. Pumps, water, pipes. Simple enough.
Well, simple in concept, but you'll see not all systems need all 3 in the way we might first think. Sponges, for example, they just use flagella to pump seawater through themselves. No real internal fluid, no heart.
Huh. Okay, so for internal circulation, the textbook points to 2 broad types.
That's right. First, open systems. Here, the fluid called hemolymph actually leaves the vessels and sort of pools in spaces called hemicoles, bathing the tissues directly. Then it finds its way back to the heart through openings called ostia.
And where do we see those?
Mostly in mollusks, think clams and snails, and in all arthropods, insects, crustaceans, spiders.
Okay, and the other type?
Closed systems. Here, the fluid now called blood stays inside continuous vessels all the way from the heart out to the body and back to the heart again.
Like our system.
Exactly. Exchange happens across the walls of tiny, often quite leaky vessels called capillaries. You find closed systems in cephalopod squid and octopus plus annelids like earthworms and all vertebrates.
Like a refrigerator's cooling system, kind of contained.
That's a good analogy. Though the source notes the distinction isn't always perfectly sharp. Even in closed systems, some fluid does leak out of capillaries.
Right. Okay, let's drill down into the fluid itself. What's actually in our blood, or in hemolymph?
Well, both are mostly plasma, that's primarily water, with lots of dissolved ions and molecules floating around. And then there are the cellular bits. In hemolymph, those are called hemocytes. They handle immunity, clotting, sometimes oxygen transport.
And invertebrates.
We have more specialized cells suspended in the plasma. You've got erythrocytes, which are the red blood cells carrying oxygen, leukocytes, the white blood cells for immunity, and thrombocytes, or platelets, in mammals for clotting.
And you can physically separate these, right? The hematocrit test.
Yeah, exactly. You spin a blood sample in a centrifuge, and the heavier cells pack down at the bottom. That packed cell volume, the hematocrit, gives you a pretty good idea of the blood's oxygen carrying capacity.
So different animals would have different values.
Hugely different. An adult Weddell seal, which dives really deep, has a hematocrit around 63.5%. Human males are around 45%. And then you get Antarctic icefish. They have less than 1%.
Wow. How do they manage?
They have no hemoglobin at all. They just rely on the oxygen dissolved directly in their very cold blood plasma. It works because cold water holds more oxygen and their metabolism is low.
Fascinating. So what about the plasma itself? beyond water and ions.
The most plentiful organic things in mammalian plasma are the plasma proteins. They make up 6–8% of plasma weight, and they're not just hanging out, they do crucial jobs.
Like what?
Well, they maintain something called colloid osmotic pressure. That basically helps keep water from leaking out of the capillaries too much. They also act as buffers, helping to stabilize blood pH.
And they vary between different animal groups.
Oh, absolutely. Arthropods have things like coagulagens for clotting and proteins involved in making their exoskeletons. Vertebrates have three main groups you might recognize. Fibrinogen for clotting. Albumins, which are the most abundant in mammals, they bind and transport stuff and contribute most to that osmotic pressure. And then globulins. These come in different types: alpha, beta, gamma. They transport things like iron, help with clotting. Some are precursors to hormones, and the gamma globulins are our antibodies.
Right, immunity. Now, speaking of transport, lipids, fats, so they don't mix with water, how do they get around in plasma?
They hitch a ride. They get packaged into lipoprotein complexes. These are basically little droplets of lipids attached to special proteins, apolipoproteins. They shuttle energy fats like triglycerides and structural fats like cholesterol and phospholipids between tissues.
And this is where we get into the famous HDL and LDL cholesterol, right?
That's exactly it. LDLs, low-density lipoproteins, deliver cholesterol and phospholipids to body cells. But if you have too much LDL, it can start accumulating in blood vessel walls, forming plaques. That's atherosclerosis, a major cause of heart disease in humans.
The bad cholesterol.
Yeah. And HDLs, high-density lipoproteins, are dubbed good because they do the opposite. They pick up excess cholesterol from cells and transport it back to the liver to be removed from the body, usually as bile.
Okay, so proteins handle fats. What about oxygen itself? How is that carried?
For that, many animals rely on specialized respiratory pigments. The most common are hemoglobins, which are iron-based. They're reddish when they bind oxygen. We have them, obviously, but so do lots of non-vertebrates.
And the other main type?
That would be hemocyanins. These are copper-based, and they actually turn bluish when oxygenated. You find them in many mollusks and arthropods like horseshoe crabs and lobsters.
Blue blood, literally.
Literally. Though it's worth noting, most insects don't bother with either. Their tracheal system, those air tubes, delivers oxygen gas directly to the tissues, so their hemolymph usually doesn't need an oxygen carrier.
And where do we find these pigments? Just floating in the plasma?
Sometimes, yes. In many annelids, the hemoglobin is just dissolved in the blood. But in many other animals, including us, they're packaged inside specialized cells, the erythrocytes, or red blood cells.
And mammalian red blood cells are pretty weird, aren't they? That biconcave disc shape, no nucleus.
They are unique compared to, say, a chicken erythrocyte, which is oval and has a nucleus. That mammalian-designed small-size, biconcave shape, no nucleus seems highly optimized.
How so?
Well, it gives a larger surface area for oxygen diffusion. The thinness means oxygen can diffuse in and out very quickly. Losing the nucleus makes more room for hemoglobin packing. And maybe most importantly, it makes them incredibly flexible, allowing them to squeeze through the narrowest capillaries found in any vertebrate group.
Almost perfectly engineered for our high metabolism.
It certainly looks that way, though it does raise a question. Birds actually have higher oxygen demands than mammals, generally speaking. So why do their red cells stay nucleated and are about twice as large?
Huh. Good question. Any answers?
The precise evolutionary reasons are still, well, a bit debated. Maybe it's linked to their faster circulation rates or more efficient lungs compensating in other ways. It's not fully resolved.
So red cells carry oxygen. Anything else?
Oh, yes. They play a vital role in transporting carbon dioxide, mainly by converting it into bicarbonate ions using an enzyme called carbonic anhydrase. Hemoglobin itself also binds some CO2 directly.
And there's more, right? Something about NO.
Yeah, this is fascinating. Hemoglobin and erythrocytes can also transport nitric oxide, NO. NO is a vasodilator. It makes blood vessels relax and widen. So, carrying NO helps ensure that oxygen-rich blood actually reaches the tissues efficiently.
That's clever.
It gets wilder. Mammalian red cells also generate tiny amounts of hydrogen sulfide, H2S, another vasodilator. That's actually the basis for the idea that garlic, which is rich in sulfur compounds, might be good for cardiovascular health by boosting H2S production.
Wow. Okay. And you mentioned an extreme example earlier, the tube worm.
Riftia, the giant tube worm living near deep-sea hydrothermal vents. Absolutely amazing. Those vents spew out toxic hydrogen sulfide. Riftia has symbiotic bacteria that use H2S for energy. Its unique hemoglobin has to transport both the toxic H2S and oxygen to its symbionts, keeping both itself and its partners alive in this extreme environment.
That's incredible adaptation. So these hardworking red blood cells,
Yeah.
how long do they last?
Not very long, actually, because mammalian and avian ones either have no nucleus or a non-functioning one. They can't repair themselves effectively. Their membranes get fragile over time. In domestic mammals, maybe 100 and 110 days, even shorter in birds.
And the spleen cleans up the old ones?
Primarily, yes. The spleen acts as a filter, removing old, damaged red cells.
Mhm.
It also serves as a reservoir for red blood cells and platelets in many mammals.
And how are they replaced? At what rate?
Through a process called erythropoiesis, which happens in hemopoietic tissues, mainly the red bone marrow in adult birds and mammals. And the rate is just staggering. In humans, it's estimated at 2 to 3 million new red blood cells produced every single second.
Wow, that's mind-boggling. And this is regulated?
Tightly regulated by a hormone called erythropoietin, or EPO. It's mostly secreted by the kidneys in response to low oxygen delivery to the tissues. If oxygen drops, EPO goes up, bone marrow makes more red cells, oxygen delivery improves, and EPO secretion goes back down.
A classic negative feedback loop.
Precisely. And it's the same ETO that some athletes unfortunately use illegally to boost their oxygen carrying capacity.
Right, okay, so we have the fluid, the cells carrying oxygen, and other things.
Mhm.
But what happens if you spring a leak? If a vessel gets damaged, how do animals stop bleeding?
That process is called hemostasis. It's absolutely crucial for survival. In vertebrates, particularly mammals, it typically involves 3 major steps.
Okay, what's step 1?
Vascular spasm. The damaged blood vessel immediately constricts. Smooth muscle in its wall clamps down to reduce blood flow through the injured area. It's an immediate reflex.
Makes sense. Minimize the loss right away. Step 2.
Platelet plug formation. Platelets, these tiny cell fragments, circulate in the blood. When a vessel is damaged, collagen fibers underneath the vessel lining are exposed. Platelets instantly stick to this exposed collagen.
They just stick.
They stick, and then they get activated. They release chemicals like ADP, which makes nearby platelets sticky too. So more platelets arrive and stick to the first layer, releasing their chemicals. It's a rapid, positive feedback cycle. You quickly build up a temporary plug at the injury site.
Positive feedback. That sounds like it could get out of control. Why doesn't the plug just keep growing and block the whole vessel?
Excellent question. The adjacent, uninjured endothelium, the lining of the blood vessel, is constantly releasing chemical signals, specifically prostacyclin and nitric oxide, NO. These signals strongly inhibit platelet aggregation.
So they act like a barrier.
Exactly. They effectively contain the platelet plug, limiting it just to the site of the injury. Very clever.
Okay, so we have constriction and a temporary plug. What's the third, more permanent step?
That's blood coagulation, or what we commonly call clotting. This is the process that transforms the blood from a liquid into a solid gel, reinforcing that platelet plug.
And this involves fibrinogen and fibrin.
That's the key event. The ultimate step is the conversion of a soluble plasma protein called fibrinogen into an insoluble thread-like protein called fibrin. This conversion is catalyzed by an enzyme called thrombin.
Thrombin makes fibrin.
Right. And these fibrin threads then spontaneously polymerize, forming a net-like meshwork that traps blood cells, red cells, white cells, platelets, reinforcing the initial plug and forming the stable blood clot.
I heard fibrin is incredibly stretchy.
It really is. The source mentions fibrin fibers can stretch to almost 3 times their length and still snap back, and over 4 times their length before breaking. That elasticity is probably important for the clot's integrity under pressure.
And thrombin seems like a key player here. Does more than just make fibrin.
It's a real multitasker. Besides converting fibrinogen to fibrin, thrombin also activates a factor that helps stabilize the fibrin mesh itself. It also acts in a positive feedback loop to promote its own formation from its precursor, amplifying the response. And it enhances platelet aggregation too, central enzyme in the whole process.
So it kicks off this whole cascade.
Exactly, the clotting cascade. It's a sequence of reactions involving about a dozen plasma proteins, mostly enzymes, called clotting factors. They're usually designated by Roman numerals. Most circulate in an inactive form.
Like dominoes.
Kind of. When the cascade is triggered either by damage to the vessel wall or contact with foreign surfaces, the first factor gets activated. That activated factor then acts as an enzyme to activate the next factor in the sequence, which activates the next, and so on.
Amplifying the signal at each step.
Precisely. One activated molecule might activate hundreds of the next. This amplification ensures that clotting can happen very rapidly when needed. Several steps in this cascade also require calcium ions and a factor released by platelets called PF3.
Why is the mammalian system so complex compared to, say, fish clotting?
Good question. The evidence, particularly from work by Russell Doolittle cited in the text, suggests this complexity evolved largely through gene duplication events. Having more steps allows for faster clotting, which is probably more critical for animals with higher blood pressures like birds and mammals.
Makes sense. Higher pressure means faster leaks.
Right. Doolittle also speculated that the added complexity might allow for finer regulatory control, adapting clotting to different needs like higher metabolic rates or specialized structures like placentas.
So the clot forms, stops the bleeding. But it's not meant to stick around forever, is it?
No, definitely not. Once the vessel wall is repaired, the clot needs to be dissolved. This is done by another plasmin protein called plasmin. Plasmin is a fibrin-splitting enzyme.
How does plasmin get activated?
It circulates as an inactive precursor, plasminogen. Tissues, especially the damaged tissue around the clot, slowly release tissue plasminogen activator, PPA. TPA converts plasminogen into active plasmin, which then starts breaking down the fibrin mesh. Plasmin is actually always active at very low levels, helping to prevent tiny, inappropriate clots from forming in the first place.
Like a continuous cleanup crew.
Sort of, yeah. And thinking about the opposite, preventing clotting that's crucial for blood-sucking parasites.
Leeches.
Exactly. Medical leeches produce a potent anticoagulant called hirudin, which directly inhibits thrombin. And get this, the Australian vampire bat produces a substance called desmoteplase. It's a plasminogen activator, like TPA, but it's hundreds of times more potent.
Wow. Nature's clot busters.
Absolutely. And these naturally evolved anticoagulants and clot dissolvers are hugely interesting for developing better drugs to treat strokes and heart attacks caused by pathological clots in humans.
What about arthropods? How do they stop leaks in their open systems?
It's a bit different but analogous. In insects and horseshoe crabs, if they get wounded, their hemolymph coagulates. They have a soluble protein called coagulagen, which gets converted into an insoluble form called coagulant, similar in principle to our fibrinogen-fibrin conversion. Hemocytes are involved, along with specialized proteins.
Any interesting connections there?
Actually, yes. One protein that barnacles use in their incredibly strong underwater glue to stick to rocks turns out to be structurally similar to mammalian clotting factor 13, which helps stabilize our fibrin clots.
That's a cool evolutionary link. And horseshoe crabs, their blood is used medically.
Yes, their hemolymph contains factors that react very strongly and rapidly to the presence of bacterial endotoxins. An extract called limulus amoebocyte lysate, or LAL, is widely used by the pharmaceutical and medical device industries to test for bacterial contamination. If bacteria are present, the extract gels up almost instantly.
Amazing. Okay, we've covered the fluid, the cells, the clotting. Let's talk about the engine, the pump. How do animals create the pressure to move this fluid?
Evolution's been quite creative here. Our source highlights at least 4 different pumping mechanisms. The simplest might be using flagella, like in sponges or even sea or urchin larvae, just whipping around to create slow current.
Okay, very basic. What else?
Then you have extrinsic muscle or skeletal pumps. This is where general body movements help propel internal fluids. Think of a sea star moving his arms. The muscles in its body wall squeeze the coalomic fluid around.
You mentioned grasshoppers earlier.
Right. A great example. When a grasshopper is actively moving, its heart, which is a simple tube, often just stops beating. The movements of its limbs and body are apparently enough to circulate the hemolymph effectively during activity.
Wow. And horses have that special pump in their hooves.
They do. The frog, that V-shaped cartilaginous plate on the sole of the hoof. When the horse steps down, the frog gets compressed, squeezing blood out of a network of veins in the hoof. When the foot lifts, elastic rebound helps draw blood back in. It's a crucial mechanism for venous return from those long, thin legs that don't have much muscle below the knee.
Clever design. What are the other pump types?
We have peristaltic muscular pumps. Think of squeezing toothpaste from a tube. A wave of contraction moves along a muscular tube. The dorsal heart of an insect works like this.
And finally, the most familiar type.
Chamber muscle pumps, or what we'd call true hearts. These are muscular chambers that contract rhythmically to expel fluid, found in most animals more complex than flatworms.
And some animals even have booster pumps.
Yes, auxiliary hearts are quite common. Cephalopods have 2 branchial hearts that specifically boost blood flow through their gills before it goes to the main systemic heart. Hagfish have several accessory hearts. Insects often have tiny auxiliary pumps at the base of their wings, legs, and antennae to ensure hemolymph reaches those extremities.
So focusing on vertebrates, how did our familiar heart evolve, from simple to complex?
It seems to have started as a pretty simple structure, basically a tube, maybe with a couple of chambers in series, like you still see in fish today. It's essentially handling one main circuit. Pump blood to the gills for oxygen, then the oxygenated blood flows out to the body, and deoxygenated blood returns to the heart.
One loop.
Right. But then some fish started breathing air, evolving lungs alongside or instead of gills. This created a problem. How to keep the oxygen-rich blood coming from the lungs separate from the oxygen-poor blood coming back from the body.
You need two circuits.
Exactly. A pulmonary circuit to the lungs and a systemic circuit to the body. This evolutionary pressure led to the gradual subdivision of the heart. You start seeing partial walls, or septa, appearing within the heart chambers in lungfish and amphibians.
To reduce mixing.
To reduce mixing. And that trend culminates in the fully divided, four-chambered hearts found in birds and mammals.
Which are essentially two pumps side by side.
That's a great way to think of it. Two pumps fused together in one organ. The right side pump receives the deoxygenated blood from the body, systemic circulation, and sends it to the lungs, pulmonary circulation. The left side pump receives the now oxygenated blood from the lungs and sends it out to the rest of the body, systemic circulation again.
And this complete separation is really important for high metabolism, right?
Absolutely crucial. It ensures that the blood going to the body tissues is fully loaded with oxygen. Problems can arise if this separation isn't perfect, or if one side of the heart struggles. The book mentions things like a sight syndrome in fast-growing broiler chickens, or brisket disease in cattle at high altitude, both related to the right ventricle struggling to pump blood through the lungs under high pressure or demand.
How do these heart chambers make sure blood flows only one way? Can't have it sloshing back and forth.
No, you definitely can't. That's the job of one-way valves. They're positioned between the chambers, atria and ventricles, and where the major arteries leave the heart. They work passively, like one-way doors, opening when pressure pushes them from the correct direction and snapping shut when pressure tries to push blood backward.
So in mammals, we have four main valves.
Yes. Two atrioventricular, or AV valves, between the atria and ventricles, and two semilunar valves, the aortic and pulmonary valves, where the aorta and pulmonary artery leave the ventricles.
And the heart muscle itself, the myocardium. What's special about it?
It's made of cardiac muscle cells, of course, which are striated like skeletal muscle, but they're arranged in spiral patterns, and importantly, they're connected end-to-end by structures called intercalated discs.
And those discs are important because
Because they contain gap junctions. These are tiny channels that directly connect the cytoplasm of adjacent cells. They allow electrical impulses, the action potentials that trigger contraction, to spread incredibly rapidly from one cell to the next.
so the whole chamber contracts together.
Exactly. It makes the entire atrium or the entire ventricle function like a single coordinated unit, what's called a functional syncytium. It means contraction is all or nothing for a given chamber. You can't have just part of the ventricle contracting. No half-hearted beats are possible.
Nice one. And cardiac muscle needs a lot of energy, right? It never stops beating.
It's an absolute energy powerhouse. Cardiac muscle cells are jam-packed with mitochondria. They can make up 30% to 50% of the cell volume. They also have lots of myoglobin to store oxygen locally. Both signs point to a heavy reliance on aerobic metabolism to constantly generate the ATP needed for contraction.
So what actually initiates that beat? Where does the signal come from?
In many animals, including vertebrates, the heart is myogenic. That means the rhythm originates within the heart muscle itself. It doesn't need external nerves to tell it to beat.
How does that work?
There are specialized cardiac muscle cells called autorhythmic or pacemaker cells. These cells have the unique ability to spontaneously and repeatedly generate their own action potentials, which then spread to the contractile cells to trigger the heartbeat.
An internal pacemaker. That seems like a good failsafe.
It's definitely an advantage. It ensures the heart keeps beating and circulating blood even if there's significant damage to the nervous system. You see myogenic hearts in vertebrates, tunicates, insects, and some mollusks and crustaceans.
But you said many animals, not all hearts are myogenic.
Correct. Some animals, notably certain crustaceans like crabs and lobsters, have neurogenic hearts. Their heart muscle requires a signal from external nerves, originating in a cardiac ganglion, to initiate contraction.
Why would that be advantageous?
It allows for more direct neural control, including the ability to completely stop the heart if necessary. Remember the grasshopper whose heart stops during activity? That's likely easier with neurogenic control, although insect hearts are generally myogenic but heavily modulated by nerves.
Okay, back to the myogenic pacemaker cells. How do they spontaneously fire? They don't have a stable resting potential.
Exactly. That's the key. Unlike regular nerve or muscle cells that maintain a steady resting potential until stimulated, pacemaker cells exhibit a pacemaker potential. Their membrane potential slowly, automatically depolarizes, drifts upwards towards the threshold potential needed to trigger a full action potential.
What causes that slow drift?
It's a complex dance of ion movements. There's an increased inward leak of sodium ions through unique channels called funny or HCN channels, a decrease in the outward flow of potassium ions, and then, as it gets closer to threshold, an increased inward flow of calcium ions through transient T-type calcium channels. Once threshold is reached, longer-lasting L-type calcium channels open, causing the rapid rising phase of the action potential. Then repolarization happens, and the slow drift starts all over again.
A continuous cycle. And how does this pacemaker signal lead to a coordinated heartbeat in the whole vertebrate heart?
It starts in a specific cluster of pacemaker cells called the sinoatrial SA node, located in the wall of the right atrium. The SA node cells have the fastest inherent rate of autorhythmicity, so they normally set the pace for the entire heart. They're the primary pacemaker.
The signal starts there, then where?
The electrical impulse generated by the SA node spreads rapidly throughout both atria, causing them to contract. It travels through specialized pathways to reach another crucial point, the atrioventricular AV node, located near the base of the right atrium.
And something important happens at the AV node. You mentioned a delay.
Yes, a critical delay. The impulse slows down considerably as it passes through the AV node, taking about 0.1 seconds.
Why is that delay so important?
It ensures that atrial contraction is fully completed and the ventricles have had enough time to fill completely with blood before the signal reaches them and tells them to contract. It synchronizes the pumping action perfectly.
Atria contract, fill the ventricles, then ventricles contract. Got it.
Right. After that brief AV nodal delay, the impulse is picked up by a specialized conduction system, the bundle of His, which splits into bundle branches, and then the Purkinje fibers that spreads the signal extremely rapidly throughout the ventricular myocardium.
Ensuring the ventricles contract forcefully and almost simultaneously.
Exactly. This coordinated, near-simultaneous contraction of the ventricular muscle fibers is essential for generating enough pressure to eject blood effectively into the aorta and pulmonary artery.
Makes sense. And if that coordination fails, you mentioned fibrillation.
Yes. If the electrical activity becomes chaotic and uncoordinated, the muscle fibers just quiver randomly instead of contracting together. That's fibrillation. Atrial fibrillation isn't immediately life-threatening, but ventricular fibrillation is disastrous because the ventricles can't pump any blood. It requires immediate electrical defibrillation.
Okay. Now, the regular contractile heart muscle cells, they have a different shape to their action potential compared to pacemaker cells, don't they? Something about a plateau.
They do. Once a contractile cell is stimulated, usually by the impulse spreading from adjacent cells, its action potential looks different. It has a very rapid depolarization phase, like nerve cells, but then it has a unique prolonged plateau phase before it repolarizes.
What causes that plateau?
It's primarily due to the slow, sustained influx of calcium ions through those L-type calcium channels we mentioned earlier. This influx roughly balances the outflow of potassium ions for a period, keeping the membrane depolarized for longer.
And why is that plateau phase so vital?
Because it makes the refractory period, the time during which the cell cannot be restimulated much longer in cardiac muscle, almost as long as the contraction itself. Meaning it's impossible to stimulate the cardiac muscle cells again quickly enough to cause summation or tetanus that's sustained, maximal contraction you can get in skeletal muscle. A tetanic contraction of the heart would be fatal. It wouldn't be able to relax and refill. The plateau prevents this.
Another built-in safety feature. You can actually visualize all this electrical activity, right, with an ECG.
Correct. An electrocardiogram, or ECG, sometimes EKG, doesn't record a single action potential, but rather the overall spread of electrical activity throughout the heart as detected by electrodes placed on the body surface.
And it shows those characteristic waves, P, Q, R, S, T.
Exactly. The typical vertebrate ECG has 3 distinct waveforms. The P wave corresponds to the depolarization of the atria initiated by the SA node firing. The QRS complex represents the depolarization of the ventricles. And the T wave signifies ventricular repolarization.
What about atrial repolarization?
It happens during the QRS complex, but the electrical signal of ventricular depolarization is so much larger that it usually masks the signal of atrial repolarization on a standard ECG.
And the timing between these waves tells you things.
Absolutely. The intervals and segments between the waves are crucial. For instance, the PR segment, from the end of the P wave to the start of the QRS, reflects that important AV nodal delay. The ST segment, between the end of QRS and the start of the T wave, corresponds to the time when the ventricles are contracting and ejecting blood, that plateau phase. Deviations from the normal ECG pattern are incredibly valuable for diagnosing a wide range of cardiac problems.
So doctors use ECGs all the time, but researchers use them on other animals too.
Oh yes, they're used clinically on horses, dogs, cats, and researchers have recorded ECGs from everything from goldfish to houseflies. It helps uncover fundamental mechanisms of heart control across the animal kingdom.
Okay, so we've got the electrical signal sorted. How does that translate into the physical pumping of blood? The mechanics.
That brings us to the cardiac cycle. This refers to the complete sequence of events that occurs during one heartbeat, involving alternating periods of contraction called systole and relaxation called diastole. We can trace it by following a drop of blood, say, through the left side of a mammalian heart.
Okay, let's follow that drop. Where do we start?
Let's start during relaxation, early ventricular diastole. At this point, both the left atrium and the left ventricle are relaxed. The pressure in the atrium is slightly higher than in the ventricle, so the AV valve, the mitral valve on the left side, is open, and blood that's returned from the lungs is passively flowing from the atrium into the ventricle. The aortic valve is closed because aortic pressure is higher than ventricular pressure.
So the ventricle is filling up passively.
Mostly, yes. Then comes late ventricular diastole. The SA node fires, triggering atrial depolarization, a P wave on the ECG. The atrium contracts, giving a final little squeeze that pushes an extra bit of blood into the ventricle, sort of topping it off. The total volume of blood in the ventricle at the very end of this filling phase is called the end diastolic volume, or EDV.
Okay, ventricle is full, now it contracts.
Now comes ventricular systole. The electrical impulse spreads through the ventricles, the QRS complex. Ventricular contraction begins, raising the pressure inside the ventricle very rapidly. As soon as ventricular pressure exceeds atrial pressure, the AV valve snaps shut.
Making the first heart sound. Lube.
That's the one, the lub. Now, for a very brief moment, both the AV valve and the aortic valve are closed. The ventricle is contracting, but the volume of blood inside isn't changing because there's nowhere for it to go yet. This is called isovolumetric ventricular contraction. Pressure skyrockets until the pressure inside the ventricle exceeds the pressure in the aorta. As soon as that happens, the aortic valve is forced open. Now blood is forcefully ejected from the ventricle into the aorta. This phase is ventricular ejection.
Does it pump out all the blood?
No, not normally. Even after a forceful contraction, there's still some blood left in the ventricle. The volume remaining at the end of systole is called the end systolic volume, or ESV. The amount of blood actually pumped out in that beat is the difference between the starting volume and the ending volume.
EDV minus ESV, that's the stroke volume.
Precisely. Stroke volume, SV, is the volume of blood ejected per beat. EDV, ESV equals SV.
Okay, ejection happens, then the ventricle relaxes.
Yes. The T wave on the ECG signals ventricular repolarization. As the ventricle relaxes, its pressure starts to fall rapidly. Once ventricular pressure drops below the pressure in the aorta, the aortic valve snaps shut, preventing backflow.
That's the second heart sound. Dupe.
That's the dupe. Now again, for another brief moment, both the aortic valve and the AV valve are closed. The ventricle is relaxing, but its volume isn't changing yet. This is isovolumetric ventricular relaxation. Pressure continues to plummet.
Until it's low enough to start filling again.
Exactly. As soon as ventricular pressure drops below the pressure in the now refilling left atrium, the AV valve is pushed open and ventricular filling starts all over again, beginning the next cycle.
Lubbed up, systole diastole, filling, ejecting. A continuous cycle.
The beautifully coordinated cycle, and the single most important measure of the heart's overall performance is its cardiac output. The total volume of blood pumped by each ventricle per minute. It's the key indicator of how well the circulatory system is meeting the body's transport needs.
And how is it calculated? You mentioned stroke volume.
Cardiac output is simply the product of heart rate, beats per minute, and stroke volume, volume pumped per beat. CO equals HRX SV.
And this can vary hugely.
Tremendously. Depending on the animal and its level of activity, the source gives examples. A resting human might have a CO around 5,000 mmol. An elite athlete during intense exercise could push that up to 35,000 or even 40,000 mmol. Compare that to a rainbow trout chilling at 10 degrees C, maybe 53 mmol. Huge range.
So how is cardiac output controlled? How can we change it so drastically?
Since CO depends on heart rate and stroke volume, the body regulates both of these factors. Heart rate is primarily controlled by the autonomic nervous system acting on the SA node.
Parasympathetic and sympathetic.
Exactly, they have antagonistic effects. The parasympathetic system, mainly via the vagus nerve releasing acetylcholine, slows down the SA node's firing rate, decreases the excitability of the AV node, lengthening the delay, and slightly weakens atrial contraction. It promotes a resting, leisurely heart rate. This is dominant at rest.
And the sympathetic.
The sympathetic system, releasing norepinephrine, does the opposite. It increases the SA node's firing rate, faster heart rate, speeds up conduction through the AV node and the rest of the production system, and also increases the force of contraction of both the atria and the ventricles. It prepares the heart for action, the fight or flight response.
So the balance between these two nerve inputs sets the heart rate according to the body's needs at that moment. What about controlling stroke volume?
Stroke volume is influenced by two main types of factors. First, there's intrinsic control, which relates directly to how much blood is returning to the heart, the venous return.
The more blood comes in, the more gets pumped out.
Essentially, yes. This is described by the Frank-Starling law of the heart. It states that the heart normally pumps out during systole the volume of blood returned to it during diastole. So, if venous return increases, the ventricle fills more, higher EDV, the cardiac muscle fibers get stretched more, and they respond by contracting more forcefully, ejecting a larger stroke volume.
Like stretching a rubber band further makes it snap back harder.
That's a pretty good analogy. More stretch leads to a more forceful contraction up to a point. This ensures that stroke volume automatically adjusts to match venous return. Interestingly, mollusk hearts show a similar response.
Okay, that's intrinsic. What about extrinsic control of stroke volume?
That comes mainly from the sympathetic nervous system again. Sympathetic stimulation not only increases heart rate, but it also directly increases the contractility of the heart muscle. This means the heart contracts more forcefully at any given end diastolic volume, leading to a greater stroke volume by decreasing the end systolic volume.
So sympathetic nerves boost both heart rate and stroke volume.
Correct. They have a powerful effect on increasing overall cardiac output. Sympathetic stimulation also constricts veins, which helps squeeze more blood back towards the heart, further increasing venous return and thus contributing to a larger stroke volume via the Frank-Starling mechanism.
It all ties together. Now, the heart muscle itself is working incredibly hard. It must need its own robust blood supply.
Absolutely. That's the job of the coronary circulation. The heart muscle is too thick, especially in active vertebrates, to get enough oxygen and nutrients just by diffusion from the blood passing through its chambers. So it has its own network of arteries, the coronary arteries, branching off the aorta right at its base, which deliver oxygenated blood directly to the myocardium.
And this supply needs to increase during exercise.
Dramatically. During increased activity, the heart is working harder and needs much more oxygen. Coronary blood flow can increase several-fold to meet this demand. This happens mainly through vasodilation widening of the coronary arterioles. This dilation is largely triggered by local metabolic changes in the heart muscle itself, particularly the release of adenosine when oxygen levels dip.
And blockages in these crucial arteries are a huge problem in humans.
Yes, coronary artery disease, leading to heart attacks, is a leading cause of death. Interestingly, the text notes that similar arterial lesions can occur in migratory salmon, possibly linked to the stress and high blood pressure associated with their long upstream journeys.
Fascinating comparison. Okay, let's shift from the pump to the pipes. How does blood actually flow through the vessels? What are the physics involved?
This falls under the realm of hemodynamics, the physics of blood flow. The fundamental relationship is quite simple, analogous to Ohm's law in electricity. The flow rate, Q, through a vessel is directly proportional to the pressure gradient across it and inversely proportional to the resistance the vessel offers to flow.
So Q equals delta P over R.
Exactly. Q EPR. Blood always flows from an area of higher pressure to an area of lower pressure. The bigger the pressure difference between the start and end of a vessel, the greater the flow rate, assuming resistance stays the same.
And resistance, what determines that?
For smooth laminar flow, resistance depends on three main things. 1, the viscosity of the fluid, how thick it is. Blood viscosity is mainly determined by the hematocrit. 2, the length of the vessel. Longer vessels offer more resistance. 3, And most importantly, the radius of the vessel.
Radius is the most important. Why?
Because resistance is inversely proportional to the radius raised to the fourth power.
The fourth power. So small changes in radius have a huge effect.
A massive effect. If you double the radius of a vessel, you decrease its resistance by a factor of 2 RO, which is 16. And since flow is inversely proportional to resistance, you increase the flow through that vessel 16-fold. Conversely, having the radius increases resistance 16-fold and cuts flow down to 1/16. This makes adjusting vessel radius an incredibly powerful way for the body to control where blood flows.
That raises that incredible question the text poses. How on earth could a brachiosaurus, with its neck perhaps 25 meters long, pump blood all the way up to its brain? The pressure needed must have been enormous.
Yeah, estimates suggest maybe 700 millimHg or even higher at the heart level. It really underscores the incredible physiological adaptations required to overcome gravity and resistance in truly giant animals. The physics are unforgiving.
So thinking about the overall layout, in closed systems, how is flow distributed from the heart to all the different organs?
A key feature is parallel branching. The large arteries leaving the heart branch out, and then those branches branch again, supplying different organs and body regions essentially in parallel, like wires connected across a battery.
Not in series, like one organ after another.
Generally not for major systemic organs. There are a few exceptions, like blood flowing from the gut to the liver, the portal system, but mostly it's parallel. This means each organ receives fresh, fully oxygenated, high-pressure blood directly from the arterial supply. It allows for independent regulation of flow to different regions.
And this fluid pressure, it's not just for transport, is it? The text mentions it could be used for other things.
Absolutely. Fluid pressure, or hydrostatic pressure, generated by the circulatory system can be harnessed for some surprising functions.
It's like movement.
Yes. Spiders famously extend their legs using hemolymph pressure, not by contracting extensor muscles, they only have flexors. Clams use hemolymph pressure to extend their muscular foot for burrowing into sand or mud. Blue crabs use fluid pressure to help move their limbs right after molting when their new exoskeleton is still soft.
What else?
Ultrafiltration. The pressure inside blood capillaries forces water and small solutes out into the surrounding tissues. This is the fundamental first step in urine formation in the kidneys.
And erection.
And erection, yes. In erectile tissues, like in mammalian genitalia or even the sensitive snout of an echidna used for probing ant nests, rapid inflow of blood under high pressure inflates the tissue, making it rigid or turgid. It's hydraulic force at work.
Fascinating versatility. Let's look briefly at some specific open systems again. Mollusks, for example.
Most mollusks, the non-cephalopod ones like snails and clams, have pretty standard open systems with myogenic chambered hearts. But even here, the flow isn't totally random or uncontrolled. Burrowing clams can selectively direct hemolymph flow to different parts of their body to help with movement. Abalone can adjust flow to different organs based on metabolic demand.
So some degree of regulation even in open systems. What about crustaceans? Lobsters, crabs?
Larger crustaceans often have surprisingly well-developed open systems. Lobsters, for instance, have a heart that pumps hemolymph into numerous arteries that branch extensively, eventually reaching capillary-sized vessels that then open into tissue spaces, lacunae. It almost blurs the line between open and closed.
And there's something about their gills.
Yes, a key feature in many crustaceans is that all the hemolymph returning towards the heart must pass through the gills first to get oxygenated before it re-enters the heart. This is an example of series flow, specifically for gas exchange, ensuring efficient oxygen uptake.
And insects. You said their system is maybe cruder.
Generally, yes. Insects typically have a simpler open system. A dorsal tubular heart that pumps hemolymph forward into the head region, from where it percolates back through the body cavity, hemicule, towards the heart, re-entering through ostia. There's less extensive arterial branching compared to, say, a lobster.
Why the difference?
The prevailing hypothesis is that because insects evolved that incredibly efficient tracheal system for delivering oxygen gas directly to the tissues via air tubes, there was less selective pressure on their circulatory system to evolve high efficiency for gas transport. Its main roles became nutrient and hormone delivery, waste removal, and immune functions.
Makes sense. The function shapes the form.
Mm-hmm.
Okay, let's swing back to vertebrates and the evolution of those circulatory pathways. We touched on the shift from 1 to 2 circuits.
Right. The basic vertebrate plan, seen in water-breathing jawed fish, is that single circuit. Heart pumps deoxygenated blood to the gills. Gills oxygenate it. Oxygenated blood flows via arteries to the body tissues. Deoxygenated blood returns to the heart. Simple loop.
But then lungs appeared.
Exactly. In air-breathing fish, like lungfish, you now have lungs as a respiratory organ, often alongside gills. This necessitates changes in the heart to start separating the pulmonary lung circuit from the systemic body circuit. You see the beginnings of septa, partial walls, dividing the atrium and sometimes the ventricle.
Trying to keep oxygenated and deoxygenated blood apart, then amphibians.
As amphibians made the transition to land, gills were often lost in adults, and they relied more on lungs and also significant gas exchange across their moist skin. Their heart typically became three-chambered, two separate atria, one receiving blood from the lung skin, one from the body, but usually only a single ventricle.
So some mixing still happens in the ventricle.
Yes, there's potential for mixing in that single ventricle, although physiological mechanisms often help minimize it.
Okay, then reptiles.
Most reptiles became fully air-breathing with lungs. Their heart generally has two atria. The ventricle is interesting. It's usually considered functionally three-chambered, but it's structurally complex, often with partial septa dividing it into subchambers. This allows for sophisticated control of blood flow.
Control like what?
Particularly in diving reptiles like turtles, snakes, and lizards, they can actually shunt blood away from the pulmonary circuit when they're underwater and holding their breath. Why send blood to the lungs if they aren't getting oxygen? They divert it to the systemic circuit instead. Very adaptive.
But crocodiles are an exception again.
They are. Crocodilians have gone all the way to a fully divided four-chambered heart, anatomically very similar to birds and mammals, with two atria and two completely separate ventricles.
But they can still shunt blood.
Yes, they have a unique connection between the major arteries leaving the heart and special valves, sometimes described as cog-like, that allow them, during a dive, to bypass the pulmonary circulation and send deoxygenated blood from the right ventricle directly into the systemic circulation, along with the oxygenated blood from the left ventricle.
Why would an ectotherm, a cold-blooded animal, evolve a four-chambered heart like endotherms, birds and mammals? Seems like overkill.
That's a great question that puzzled biologists for a long time. The thinking now, based partly on fossil evidence, is that the ancestors of modern crocodilians might have been much more active, possibly even endothermic terrestrial predators.
Really? Like warm-blooded crocs?
Maybe. And some genomic evidence seems to support this idea too. Alligator mitochondrial DNA, for example, seems to have evolved at a rate more similar to mammals than to other reptiles, which could hint at a past period of higher metabolic rate, maybe endothermy. The four-chambered heart might be a retained feature from that ancestral condition.
Fascinating evolutionary detective work. So that leaves birds and mammals.
Birds and mammals, with their consistently high metabolic rates, endothermy, have independently evolved completely separate four-chambered hearts. No possibility of shunting or mixing. All blood returning from the body goes to the lungs. All blood returning from the lungs goes to the body. Maximum efficiency for oxygen delivery.
And you mentioned earlier that a single gene might be key to this heart division.
Yes. Research highlighted in the text points to a regulatory gene called TBX5. It codes for a transcription factor, a protein that controls the activity of other genes. The pattern of where TBX5 is expressed in the developing heart embryo seems to correlate precisely with whether and how the septum, the wall dividing the chambers, forms in different vertebrate groups.
So changes in the regulation of this one master gene could have played a huge role in driving the evolution from a two-chambered to a four-chambered heart.
It certainly looks like a major player. It's a fantastic example of how evolution can work through changes in developmental control genes to produce large-scale anatomical innovations. It gives us insight right down to the molecular level of how these crucial structures evolved.
Amazing. You also mentioned that some organs, like the digestive system or kidneys, can tolerate temporary reductions in blood flow. Why them specifically?
These are sometimes called reconditioning organs. At rest, they typically receive a blood flow far greater than what they need just for their own metabolic survival. They're receiving blood to process it, absorb nutrients, filter waste, etc. So during exercise or stress, blood flow can be safely diverted away from them temporarily towards muscles or the brain without causing immediate damage.
But the brain and heart can't tolerate that.
No, they are highly dependent on a continuous high level of blood flow and oxygen delivery. Their own metabolic needs are very high, and they have little capacity for anaerobic metabolism. In mammals, even a few minutes of oxygen deprivation can cause irreversible brain damage. The heart muscle also suffers quickly without adequate coronary flow.
Okay, So just to quickly recap, the vessel types in a closed system like ours, starting from the heart.
Sure. Blood leaves the heart via large arteries. These branch repeatedly into smaller arteries, eventually leading to arterioles. Arterioles are the primary sites of resistance control, determining flow into specific capillary beds.
And capillaries are where the action happens.
Capillaries are the microscopic vessels where the actual exchange of gases, nutrients, and wastes between blood and tissues occurs. Capillaries then merge to form small venules. Venules join together to form larger veins, which finally return the deoxygenated blood back to the heart.
Arteries, arterioles, capillaries, venules, veins, and that arterial capillary venule section is the microcirculation.
Correct. That's the microcirculation, where the key functions of exchange and local flow regulation take place.
Let's look closer at arteries. You said they're rapid transit passageways, but also act as a pressure reservoir. How does that work?
Yes, both are key roles. Because arteries have relatively large diameters, they offer very little resistance to blood flow, so blood can move through them quickly away from the heart. But importantly, their walls are thick and highly elastic. They contain lots of collagen fibers for strength to withstand high pressure, and elastin fibers for stretchiness.
Like a giraffe's arteries must be really strong.
Exactly. They have to handle immense pressure. So when the ventricle contracts, systole, and ejects blood into the arteries stretch or distend because more blood is flowing in than is flowing out into the downstream arterioles at that moment.
They bulge outwards, storing energy.
Precisely. They store some of the pressure energy from the heart's contraction in their elastic walls, like blowing up a balloon. Then, during diastole, when the ventricle is relaxing and refilling, the stretched arterial walls passively recoil, squeezing down on the blood inside.
And that keeps the blood moving forward.
Yes, it maintains a continuous driving pressure and keeps blood flowing smoothly out into the arterioles and capillaries even when the heart itself isn't actively pumping. This elastic recoil is why arterial blood pressure doesn't drop to zero between heartbeats.
And that gives us our systolic and diastolic pressure readings.
That's exactly it. Systolic pressure is the peak pressure reached in the arteries during ventricular ejection. For example, typically around 120 millimole Hg in a healthy young human. Diastolic pressure is the minimum pressure remaining in the arteries just before the next ventricular contraction when the walls are recoiled, for example, around 80 millimole Hg.
And the average pressure driving flow.
That's the mean arterial pressure, MAP. It's not just a simple average of systolic and diastolic because the heart spends more time in diastole. A good estimate is MAP at diastolic pressure plus 13, systolic pressure, diastolic pressure. For our 2080 example, MAP would be about 93 millimole Hg. This is the average pressure actually pushing blood through the tissues over the whole cardiac cycle.
And these pressures vary wildly across animals, right? You mentioned a jumping spider.
Yeah, the source notes a jumping spider can have systolic pressures maybe up to 400 mHg, presumably needed for their hydraulic leg extension. Compare that to a trout, maybe around 43 mmHg. It's all tailored to the specific physiological challenges and lifestyle of the animal.
Okay, downstream from the elastic arteries are the arterioles, much smaller, and you said they are the main resistance vessels.
Yes, arterioles are critically important. They are the major site of resistance in the entire vascular tree. Their walls have a thick layer of smooth muscle that can contract or relax, significantly changing the vessel's radius.
And because resistance is so sensitive to radius?
Adjusting arteriolar radius allows the body to do two crucial things. One, control the distribution of cardiac output. By selectively constricting or dilating arterioles supplying different organs, the body can direct more blood flow to active tissues and less to inactive ones. 2. Help regulate overall systemic arterial blood pressure. Widespread changes in arterial resistance have a major impact on MLP.
And because they offer so much resistance, the pressure drops a lot as blood flows through them.
Yes, there's a significant pressure drop across the arterioles. This also means that the pulsatile pressure fluctuations seen in the arteries, systolic, diastolic, are largely dampened out. By the time blood reaches the capillaries, the flow is much smoother and non-pulsatile.
So these arterioles have smooth muscle they can constrict or dilate?
Right. Vasoconstriction is narrowing, increases resistance, reduces flow, and vasodilation is widening, decreases resistance, increases flow. Arterioles normally maintain a baseline level of partial constriction called vascular tone. This is important because it allows them to either dilate further or constrict further, giving a wide range of control.
So how does the body decide which arterioles constrict and which dilate? How is blood flow directed?
It's based on need, regulated by both local and extrinsic factors. Remember, blood is delivered to all the parallel capillary beds at roughly the same mean arterial pressure, so the amount of flow an organ receives depends almost entirely on the resistance of its specific arterioles. If an organ arterioles dilate, low resistance, it gets more flow. If they constrict, high resistance, it gets less flow.
Like adjustable valves on parallel pipes. What are the local controls?
Local intrinsic controls are factors originating within the tissue itself that directly affect the smooth muscle of its arterioles. These are primarily aimed at matching blood flow to the tissue's immediate metabolic needs. For example, if a muscle becomes highly active, it produces more CO2, lactic acid, and uses up oxygen. These local chemical changes directly cause vasodilation of the muscle's arterioles, increasing blood flow to deliver more oxygen and remove waste.
The tissue calls for what it needs. What about extrinsic controls?
Extrinsic controls are factors originating outside the organ, mainly the sympathetic nervous system and certain hormones like epinephrine, vasopressin, angiotensin. These are generally concerned with regulating the needs of the whole body, especially maintaining overall arterial blood pressure.
So sympathetic nerves can cause widespread vasoconstriction.
Mostly, yes. Sympathetic stimulation typically causes vasoconstriction in arterioles supplying organs like the digestive tract, kidneys, and skin. This helps maintain or increase overall blood pressure. However, in some crucial areas, the response is different.
Like in active muscles.
Exactly. Arterioles in skeletal muscle and heart muscle can actually dilate in response to sympathetic stimulation via different receptors or overriding local factors during exercise. This, combined with vasoconstriction elsewhere, helps divert a larger fraction of the cardiac output to the working muscles.
A coordinated redirection of flow.
It's a beautifully integrated system. During exercise, local factors in the muscles cause vasodilation there, while sympathetic nerves cause vasoconstriction in the gut and kidneys. This shunts blood to where it's needed most.
But doesn't all that dilation in the muscles risk dropping overall blood pressure?
That's the critical balance. While resistance in the active muscles drops significantly, the widespread vasoconstriction in other large vascular beds, like the gut and skin initially, helps to keep total peripheral resistance from falling too drastically. Plus, cardiac output increases significantly. The net effect is usually a moderate increase in mean arterial pressure during exercise, ensuring adequate driving pressure for all tissues, especially the brain.
And the brain's blood flow is protected.
Yes, cerebral blood flow is kept remarkably constant, largely through its own powerful local autoregulation, relatively independent of sympathetic control. The cardiovascular control center in the brainstem, medulla, constantly integrates signals about pressure, oxygen levels, CO2 levels, and activity levels to orchestrate these adjustments.
Amazing coordination. So the arterioles act as gatekeepers, controlling flow into the capillaries, and the capillaries are where the real exchange happens.
They're the ultimate functional units, yes. Everything else, heart, arteries, veins, exists primarily to serve the capillaries. Their entire structure is optimized for efficient diffusion between blood and the surrounding interstitial fluid bathing the cells.
How are they optimized? Based on Fick's law of diffusion.
Precisely. Fick's law tells us diffusion rate depends on the concentration gradient, the surface area A, the permeability or diffusion constant D, and inversely on the diffusion distance. Capillaries maximize or minimize these factors perfectly.
Okay, how do they minimize distance?
Capillary walls are incredibly thin, just a single layer of flattened endothelial cells. Plus, capillaries are so narrow that red blood cells often have to squeeze through in single file, bringing oxygen very close to the capillary wall. And capillaries form such dense networks that almost no cell in the body is more than about 0.1 millimeters away from one, tiny distance for diffusion.
Okay. Maximize surface area A.
The sheer number of capillaries is astronomical. Estimates range from 10 to 40 billion in a human body. Laid end to end, they'd circle the Earth multiple times. Their total surface area for exchange is enormous, maybe 600 square meters, even though at any given moment, only about 5% of your total blood volume is actually within the capillaries.
Like spreading a tiny amount of paint over a huge area.
Exactly. Maximizes contact. And finally, maximizing permeability, D. Capillary walls aren't completely sealed. Between the endothelial cells are narrow, water-filled pores, or clefts that allow water and small water-soluble substances, like ions, glucose, amino acids, to pass through relatively easily. Lipid-soluble substances, like oxygen and CO2, can diffuse directly through the endothelial cell membranes themselves.
Do all capillaries have the same size pores?
No, permeability varies depending on the organ. Brain capillaries have very tight junctions between cells, forming the protective blood-brain barrier that limits passage of many substances. Liver capillaries, on the other hand, have large gaps, making them very leaky, even allowing proteins to pass through easily.
Tailored to the organ's function, so exchange happens down concentration gradients.
Absolutely. The circulatory system works constantly to maintain favorable gradients. Blood arriving at the systemic capillaries is high in oxygen and nutrients, and low in CO2 compared to the tissue cells. So oxygen and nutrients diffuse out of the blood into the interstitial fluid and then into the cells. Conversely, cells produce CO2 and other wastes, making their concentration high, so these diffuse into the interstitial fluid and then into the capillary blood to be carried away.
And capillaries can regulate flow within their own beds. That's something about sphincters.
Yes, many capillary beds have precapillary sphincters. These are little rings of smooth muscle located right where a capillary branches off from an arteriole. They act like tiny taps or stopcocks. They're highly sensitive to local metabolic conditions. If a tissue area is inactive, its precapillary sphincters tend to constrict, reducing or stopping blood flow through those specific capillaries. If the tissue becomes active, the local chemical changes cause the sphincters to relax, opening up the capillaries and increasing perfusion.
Fine-tuning flow right down to the micro level. Now, for all this exchange to happen effectively, blood flow must slow down dramatically in the capillaries, right?
Yes, and this relates to an important distinction. Flow rate versus velocity of flow. The flow rate, the volume of blood passing a point per unit time, for example, ml mean, must be the same in all parts of the circulatory system in series. It's equal to the cardiac output. If 5 liters per minute leave the heart, 5 liters per minute must flow through the aorta, 5 liters per minute through all the arteries combined, 5 liters per minute through all the capillaries combined, and so on.
Conservation of flow. But velocity is different.
Velocity, the linear speed at which the blood is moving, for example, on the sec, depends on the flow rate divided by the total cross-sectional area of all the vessels at that level. Velocity equals flow rate total cross-sectional area.
And the capillaries, although individually tiny, have a huge total cross-sectional area when you add them all up.
By far the largest total cross-sectional area in the entire system, much larger than the aorta or all the arteries combined. So even though the flow rate is the same, because that flow is spread out over this enormous area, the velocity of blood flow becomes very, very slow in the capillaries.
Like a river widening into a lake.
Perfect analogy. The river, aorta, flows fast. It widens into a huge, slow-moving lake, the capillary beds. Then it narrows again into another river, the veins, and the flow speeds up again. This slow transit time through the capillaries, maybe 1-2 seconds, is crucial. It provides enough time for that vital diffusion and exchange to occur.
And all this exchange is mediated by the fluid between the capillaries and the cells.
Yes, the interstitial fluid. This is the fluid that fills the spaces between cells. It's the true internal environment that directly bathes almost all cells in the body. Plasma filters out of the capillaries, becoming interstitial fluid. Substances diffuse through it to reach the cells. Waste products diffuse from cells into it. And then most of it returns to the plasma or enters the lymphatic system. It's like the intermediary communication medium.
The source called it the evolutionary descendant of an open system.
In a way, yes. In animals with closed circulation, the interstitial fluid compartment represents what remains of that direct bathing fluid found in open systems. It actually makes up about 80% of the total extracellular fluid volume in mammals.
And fluid moves between plasma and interstitial fluid, not just by diffusion, but by bulk flow.
Yes. Besides diffusion of individual solutes, there's also bulk flow of water and small solutes across the capillary wall, driven by pressure differences. Fluid filtering out of the capillary into the interstitial space is called ultrafiltration. It's driven mainly by the capillary blood pressure being higher than the interstitial fluid pressure. Fluid moving back into the capillary from the interstitial space is called reabsorption, driven mainly by the colloid osmotic pressure of the plasma proteins.
So filtration out, reabsorption back in, does it balance perfectly?
Almost, but not quite. Typically, slightly more fluid filters out than is reabsorbed back into the blood capillaries each day.
So where does that excess fluid go? Does it just accumulate?
That's where the lymphatic system comes in. It's like a secondary drainage system, an accessory route for fluid return.
Okay, tell me about the lymphatic system.
It's a network of one-way vessels that originates as tiny blind-ended tubes called initial lymphatics or lymphatic capillaries permeating almost all tissues. These vessels collect the excess interstitial fluid, along with any leaked plasma proteins and other large particles like bacteria or cell debris that couldn't get back into the blood capillaries.
How does the fluid get into these lymphatic vessels?
The walls of the initial lymphatics are made of endothelial cells that overlap slightly, forming flap-like mini-valves. When the pressure in the interstitial fluid gets higher than inside the lymphatic capillary, these flaps are pushed inwards, opening like swinging doors and allowing fluid, now called lymph, to enter. When pressure inside is higher, the flaps are pushed shut, preventing backflow.
Clever one-way entry. And how does the lymph then move through the system and get back to the blood?
Unlike the blood circulation, there's no central pump for the lymph. In some vertebrates, like amphibians and reptiles, there are pulsating lymph hearts. But in mammals and most adult birds, lymph flow is propelled mainly by two things. One, the smooth muscle in the walls of the larger lymphatic vessels contracts rhythmically an intrinsic lymph pump. Two, contractions of surrounding skeletal muscles squeeze the lymphatic vessels, pushing lymph along.
And they must have valves, too.
Yes, larger lymphatic vessels have numerous one-way valves, similar to veins, ensuring that lymph always flows in one direction, eventually returning to the blood circulation, typically emptying into large veins near the heart.
So its main job is fluid return.
That's a primary role, preventing edema or swelling. But the lymphatic system has other crucial functions, too. It's essential for immune defense. As lymph percolates through lymph nodes located along the vessels, it's filtered, and immune cells, lymphocytes, monitor it for pathogens, initiating immune responses.
And something about fat transport.
Yes, that's the third major function. Digested fats are absorbed in the small intestine mostly as large particles called chylomicrons. These are generally too large to enter the blood capillaries directly. Instead, they enter the initial lymphatics, called lacteals in the gut, and are transported via the lymphatic system into the bloodstream.
Okay, so lymph handles the overflow and plays roles in immunity and fat absorption.
Yeah.
Now, let's complete the blood circuit. Back to the heart via venules and veins.
Right. Capillaries merge into venules, which are small return vessels. Venules then converge to form progressively larger veins. These are the main conduits for returning deoxygenated blood in the systemic circuit back to the heart.
How are veins different from arteries?
Structurally, their walls are thinner and less muscular than arteries of comparable size. They operate under much lower pressure. Functionally, because they are thin-walled and stretchy, distensible, they act as a significant blood reservoir.
They hold a lot of blood.
Yes. At any given time, up to 60% or even more of your total blood volume might be residing in the systemic veins. Their capacity can change significantly. If the body needs to increase the circulating blood volume, sympathetic stimulation can cause venous vasoconstriction, squeezing the stored blood out of the veins and increasing venous return to the heart.
So blood pressure in veins is low. How does blood manage to get back to the heart, especially from the lower body, against gravity?
That's a key challenge. Several factors contribute to venous return. One, that sympathetically induced venous vasoconstriction we just mentioned helps push blood forward. Two, the skeletal muscle pump is crucial. When you contract your leg muscles, for example, they compress the deep veins embedded within them, squeezing blood upwards towards the heart.
Like milking the veins.
Exactly. And this is where one-way venous valves are absolutely essential. These valves, located frequently within limb veins, prevent the squeezed blood from flowing backward when the muscles relax. They ensure flow only goes towards the heart. This is especially important in tall animals like giraffes.
So valves stop backflow, muscles pump it forward. Anything else?
Respiratory activity also helps. When you inhale, pressure changes in the chest cavity help draw blood towards the heart. And there's even a slight suction effect from the heart itself during ventricular relaxation. But the muscle pump and valves are the main players, especially for overcoming gravity.
Okay, we've journeyed through the fluid, the pump, the pipes, the exchange sites, the drainage system, and the return routes. How does this all work together as an integrated system? What are the main goals?
The cardiovascular system is constantly juggling demands to meet two primary, sometimes conflicting goals. The absolute top priority for short-term survival is proper gas and heat transport.
Getting oxygen in, CO2 out, and managing temperature.
Right. At rest, the system is generally regulated homeostatically to ensure consistent oxygen delivery, particularly to vital organs like the brain, heart, and kidneys. During activity or environmental challenges, cardiac output needs to be adjusted, often increased significantly to boost flow to working muscles and maybe to the skin for heat dissipation.
And the second major goal?
That's maintaining adequate arterial blood pressure. This is also regulated, though perhaps more flexibly than, say, body temperature. Pressure needs to be high enough for several reasons.
Such as?
To overcome gravity and push blood through the resistance of the vessels, ensuring sufficient flow, especially to the brain. If your diastolic pressure drops too low, say below 50 mmHg, blood might literally not reach your brain when standing up. Pressure is also needed to drive ultrafiltration in the kidneys for urine formation.
But done too high.
Exactly. Pressure shouldn't be so high that it creates unnecessary work for the heart muscle or increases the risk of damaging blood vessels, like causing an aneurysm or stroke.
So transport needs versus pressure maintenance. What if they conflict?
Generally, the need for adequate transport, especially oxygen delivery, seems to win in the short term. The source gives the example of hypertension, high blood pressure. Often the body might be raising pressure partly as a way to maintain adequate cardiac output and tissue perfusion, even though the high pressure itself might be harmful long term. Or consider regulating body temperature. If you're overheating, your body will dilate blood vessels in the skin to lose heat, even if this causes your blood pressure to drop somewhat. Temperature regulation can override pressure regulation.
Interesting priorities. How does the body coordinate all this during something common like exercise?
Okay, let's unpack that. During strenuous activity, like running, several major cardiovascular adjustments happen simultaneously and are tightly coordinated.
What are the key changes?
Two main things. One, a substantial increase in cardiac output. Both heart rate and stroke volume go up significantly, pumping much more blood per minute. Two, a major redistribution of that blood flow. There's massive vasodilation in the active skeletal muscles, the heart muscle itself, and often the skin to get rid of heat. This dramatically increases blood flow to these areas. At the same time, there's vasoconstriction in organs like the digestive tract and kidneys, diverting blood away from them.
So more blood overall, and it's sent where it's needed most.
Precisely. More oxygen and fuel delivered to working muscles, waste products removed faster, excess heat dissipated via the skin. And remarkably, despite the huge increase in cardiac output, the overall mean arterial pressure usually only increases modestly.
Why only modestly?
Because the massive vasodilation in the muscles causes a large decrease in total peripheral resistance, TPR, which largely offsets the effect of the increased cardiac output. Remember MAP-COX TPR. It's a beautifully balanced response.
And this involves both local and central control.
Yes. Local factors, like CO2, acid, low O2, in the muscles drive much of the vasodilation there. But the overall response, increased cardiac output, vasoconstriction elsewhere, is orchestrated by the sympathetic nervous system under the command of the cardiovascular control center in the brainstem.
And you mentioned anticipation.
Yeah, that's truly fascinating. Higher brain centers, like the motor cortex planning the movement, can actually send signals down to the cardiovascular control center before the activity even starts. This can trigger some of these adjustments, like increased heart rate in anticipation, priming the system for the expected increase in demand. Even just thinking about exercise or feeling stressed can initiate parts of the fight-or-flight cardiovascular response.
Preparing for action before it happens. What about long-term blood pressure regulation, not just moment to moment?
Long-term control of blood pressure is intimately linked to regulating total blood volume. This primarily involves controlling the body's salt and water balance, which is largely the job of the kidneys under the influence of hormones like renin, angiotensin, aldosterone, and vasopressin, also called antidiuretic hormone, ADH.
So hormones in kidneys manage volume, which affects pressure long-term.
Exactly. Maintaining blood pressure over days, weeks, and years depends heavily on matching fluid intake and output to keep blood volume stable. So you have this constant interplay. Moment-to-moment adjustments in cardiac output and peripheral resistance handle short-term fluctuations, while long-term regulation hinges on blood volume control via hormones and the kidneys. All three factors, cardiac output, total peripheral resistance, and blood volume, are continuously juggled to maintain a relatively stable blood pressure while meeting the body's ever-changing transport needs.
It's an incredibly dynamic and complex system. From the tiny pores on a capillary wall allowing exchange to the incredibly powerful beat of a four-chambered heart pushing blood against gravity. It's really clear that animal circulatory systems are just masterpieces of biological engineering. They're designed to overcome the very basic limits of physics.
They really are a testament to evolution's ingenuity, providing not just transport, but as we saw, also ways to transmit force and layers upon layers of intricate regulation that link everything together from the molecular level, like that TBX5 gene, right up to the whole organism's survival in its environment.
So what does this all mean for you listening? Well, next time you feel your pulse quicken when you exercise, or maybe even just watch a spider jump, perhaps take a moment to appreciate that hidden, complex dance of fluids, pumps, and vessels working tirelessly, nonstop, just beneath the surface, keeping life flowing. It really shows there's always more to learn about the world around us and inside us.
Absolutely. And thinking critically about how these systems work, how they evolved, it's essential in navigating the sheer amount of information out there. Keep asking those why and how questions.
We really hope this deep dive into animal circulation left you feeling not just, informed, but genuinely curious to explore even further.